This crazy looking thing is the TRICARBOXYLIC ACID cycle. This is part 2 of burning sugar for energy. You start with glycolysis, you turn glucose into pyruvate, you process pyruvate into acetyl CoA, and you hook acetyl CoA up with oxaloacetate to make citrate. Now you have citrate, you can start spinning this cycle and you can start reducing NAD plus and FAD into NADH and FADH2. These are high energy electron transport molecules, and they are going to dump their high energy electrons into the electron transport chain inside of your mitochondria, and they're gonna start making a ton of energy, a ton of ATP for your body. So in today's video, all we're doing is going over the metabolic pathway, the biochemical pathway of the tricarboxylic acid cycle. And there goes a motorcycle. Brum, brum. Okay, tricarboxylic acid cycle. How the frick does this thing even work? Well, it starts with the whole reason this cycle can spin is because of the 6-carbon molecule citrate. Citrate comes from oxaloacetate plus acetyl CoA. Acetyl CoA is a 2-carbon molecule, oxaloacetate is a 4-carbon molecule, and the enzyme Citrate Synthase hooks up that acetyl CoA with oxaloacetate, spits out coenzyme A, and it gives us citrate, a 6-carbon molecule. Next, the enzyme Aconitase turns citrate into isocitrate, and it uses a water molecule to kind of rearrange citrate into isocitrate. This enzyme, Isocitrate Dehydrogenase, is critically important to this whole entire metabolic pathway because it is giving us NADH. So isocitrate dehydrogenases, isocitrate dehydrogenase, dehydrogenases isocitrate, and it turns that into alpha keto glutarate. When isocitrate dehydrogenase is doing its little job, it's spitting out a carbon dioxide. It's spitting out a CO2. So isocitrate is a 6-carbon molecule. Alpha keto glutarate only has 5 carbons. One of those carbons is lost as CO2. Also, this enzyme uses NAD plus to facilitate the oxidation of isocitrate. So NAD plus comes in, isocitrate is oxidized, and NADH reduced, comes out. Now we have alpha keto glutarate, and the enzyme alpha keto glutarate dehydrogenase pretty much does the exact same thing. So here we go from alpha keto glutarate, a 5-carbon molecule, to Succinyl CoA, a 4-carbon molecule. NAD plus comes into this reaction, and it comes out as NADH reduced. So this is alpha keto glutarate dehydrogenase. It oxidizes alpha keto glutarate into Succinyl CoA, and because it is being oxidized or it's being dehydrogenated, that hydrogen is stuck onto NAD plus, and we spit out NADH. This is also a step where we reduce our carbon numbers. This step also makes CO2. So we go from 5-carbon alpha keto glutarate to 4-carbon Succinyl CoA, and we spit out a carbon dioxide. So right here, we've lost 1 carbon dioxide, and right here, we've lost 1 carbon dioxide. We've gone from 6-carbon to 5-carbon, now to 4-carbon at Succinyl CoA. What alpha keto glutarate dehydrogenase also does is it pops this CoASH, coenzyme A, with a thiol group onto alpha keto glutarate, and it gives us Succinyl CoA, Succinyl CoASH. This next enzymatic reaction yields us an ATP molecule, and that is because this SH, this thiol group, it has a high amount of potential energy. So once we break that bond here, we are actually able to use that energy and phosphorylate ADP into ATP. So right here, this is the enzyme Succinyl CoA Synthetase. A synthetase is any enzyme that is dealing with ATP as well. So Succinyl CoA, 4 carbons into succinate, and it makes 1 ATP and it releases that CoASH, coenzyme A. Now we have succinate, and we use the enzyme Succinate Dehydrogenase to make FADH2. FAD shows up right next to succinate. The enzyme Succinate Dehydrogenase processes succinate, spits out FADH2. This FADH2 is going to go to the electron transport chain and dump its electrons there. Now we have the enzyme, or the molecule fumarate, still a 4-carbon molecule, and we're going to use the enzyme fumarase to add water and turn fumarate into malate. Malate is a 4-carbon molecule. Now we have malate. We use malate dehydrogenase to oxidize malate into oxaloacetate. Malate dehydrogenase has NAD+, and it spits out an NADH. So that NAD+ is facilitating the oxidation of malate into oxaloacetate. This NADH is going to go dump its electrons now into the electron transport chain, and it's going to help us make a ton of energy. Now we have oxaloacetate. We cannot spin this cycle again until we have citrate. This is where sugar is really important, because this acetyl CoA comes from pyruvate. Pyruvate comes from glucose from the process of glycolysis. So you eat sugar, sugar enters your cell, you send that sugar down the metabolic pathway of glycolysis, and you get pyruvate. Pyruvate is then processed into acetyl CoA. Acetyl CoA now combines with oxaloacetate to make citrate. As soon as you have that citrate, you can start spinning this cycle over and over and over again, and see all these things literally just like flying off the cycle. This NADH, NADH, FADH2, NADH. These are all high energy electron transport carriers, and they are dumping their high energy electrons into the electron transport chain. As those electrons are pumped along the chain, they are creating a strong electrochemical gradient which stores potential energy. That potential energy is used to phosphorylate ADP into ATP, and that is where your body is making so much energy. So the whole reason this cycle is spinning is because glucose showed up in your cell, or fatty acids showed up in your cell, and your body's decided, let's burn these molecules for energy, let's start spinning this cycle. That is literally it. That is the tricarboxylic acid cycle. That is what your body does when it is burning sugar or fats for energy, and it is sending those fuel molecules through the tricarboxylic acid cycle to access their high energy electrons, send them down the electron transport chain, and make so much ATP. There's a bunch of hormonal regulations to this cycle. So insulin can stimulate it, insulin hormone will turn it up, glucagon will slow it down, and then calcium also has a role in controlling that cycle. But insulin is your hormone of energy abundance. So much sugar came in, so much fat came in. Let's increase these cycles that burn with our abundance of fuel. Glucagon slows down that cycle. Glucagon is a hormone of fuel depletion. Glucose is gone, glucagon. So it's slowing down the cycle, and it's saying like, hold on to everything. Let's preserve glucose and preserve our fatty acids. Let's not burn them because we're kind of low on fuel molecules. So let's save what we have right now. If you want to learn the specifics on how insulin and glucagon are controlling glycolysis and your body's energy making sugar burning cycles, see my video on the hormonal regulations of glycolysis, and you'll figure all of that out. Okay, everybody, if you got any value out of this video whatsoever, like, comment, and subscribe. It makes a huge impact on the quality of video I'm able to produce for you, and it helps me put more time, energy, and resources into making these videos for you guys. So I hope you all have such a rocking day, and I'll see you in the next video all about the electron transport chain. Also, a little bit of behind the scenes. Here I am in Mexico. Here are my notes on the tricarboxylic acid cycle, and here's exactly what you're learning in today's video, and I hope you all have a rocking day. See you later.